Hello everyone, my name is Qingjin Li from Stevens Institute of Technology. Today I will talk about carrying nanoparticles through porous media using foam for contaminant site remediation. This work is done with my advisor, Dr. Valentina Prijob. This presentation is divided into 5 parts. So firstly, I will start with motivation and introduction and introduce the mathematical model of foam transport in the presence of nanoparticles. Then I will describe the setup and the experimental approach with the results and the simulations. Finally, I will summarize and draw the conclusions. To address the soil contamination problem, several remediation techniques have been developed over the years, such as thermal remediation, bioremediation, chemical remediation, air sparging, nanoremediation. However, these methods have limitations like the cost due to chemicals and water consumption, the inefficiency due to soil heterogeneity, and the back diffusion. Foam flooding developed for soil for enhanced oil recovery has been proposed as a method to overcome soil heterogeneity while minimizing water consumption. If the foam is stabilized with nanoparticles, it could be combined with nanoremediation, allowing more efficient delivery of reactive nanoparticles. So in this presentation, I will talk about a novel process which consists in combining foam flooding with nanoremediation. The objective is to better control nanoparticle mobility due to the high foam viscosity, as well as reduce water consumption. On the left-hand side, a schematic diagram of bubbles and nanoparticle transport in porous media is shown. In brown is a soil, in blue is a liquid, and in white is a gas. So the zone with in 2 bubbles is named as laminar, and it is here where the surfactant and the surfactant with nanoparticle sit. So 2 main questions in our research are which is the transport behavior of nanoparticles in the presence of foam in porous media, and what is the delivery efficiency of nanoparticles by foam. To answer these 2 questions, we developed a mathematical model to describe the transport of foam in porous media and in the presence of nanoparticles, and calibrated with experiments. So the first part of the model is foam transport. It is composed of a fraction flow equation, which is used to represent the flow of water and gas phase. And then the population balance equation, which means to describe the foam generation and the corruption process. And F in here is the bubble concentration, which means the number of bubbles per unit volume. RGRC here is the generation rate of foam, and RC is a corruption rate of foam. And these 2 terms controls the creation and the loss of foam. So according to other studies, the rate of generation of pure foam could be represented as the power law, power relationship with pressure gradient. Based on our early work, this relationship still holds for foam generation in the presence of nanoparticles. So the rate of corruption is, and also the rate of corruption is assumed as a function of water saturation. So with this, we could determine the number of bubbles in the porous media, and then considering its effect on the flow by the effective viscosity. It is a crucial part to demonstrate the mobility control. Here is a specific case showing the effective viscosity of foam. The picture below shows the flow through a saturated microchip. On the left-hand side is a gas was injected into the water saturated chip, where on the right-hand side is a gas was injected into the surfactant solution saturated chip. So if only single phase gas is injected, it will create a preferential flow path along the boundaries Therefore, the liquid in the middle area of the chip will not be displaced. However, if the chip inside has a surfactant, the foam will be generated during the this drainage process. Then it will have a stable front of foam, and which could swap the water all across the chip. And the next part is nanoparticle transport. The first equation describes the transport of nanoparticles within the liquid phase, where mechanisms of advection and desorption onto the porous medium and surface occur. And the second equation describes the nanoparticle fraction, which travels with the gas phase upon adsorption onto the gas-liquid interface. The equation here at the bottom is the population balance equation describing the evolution of nanoparticle size distribution within the liquid phase by accounting only for aggregation. Here, the nanoparticles is classified into groups of different sizes. On the left-hand side of the equation, it is the rate of change of number of a specific group. On the right-hand side is the first term is the formation of this group, while the second term is the loss of the group due to aggregation of nanoparticles. So then it goes to the beta function. It is actually the aggregation kernel, which depends on the the interaction force between particles, like 1/2 force, electrostatic force, and the magnetic force. After getting the size of aggregate nanoparticles, it could be used to calculate the attachment and detachment coefficient. On the right-hand side, it is the equation describing the attachment and detachment process, and to calculate the corresponding coefficient. The model contains several parameters that we have to estimate it by running systematic experiments. So here is the setup we use to run the experiment. And during each experiment, the column was saturated with water or surfactant solution. Upon stabilization, nitrogen and a solution containing either surfactant or surfactant nanoparticles were co-injected. Then the samples were collected and characterized using a UV spectrometer to measure the nanoparticle concentration in the effluent. So the system was continuously monitored with the temperature and the pressure probes. So here is the experimental results for a foam stabilized with surfactant. The black line is the model upon fitting. Here, the model, the foam quality was changed as 86%, 71%, and 57%. The top row shows the pressure drop of the column, and while the bottom part is the remaining water saturation as a function of per volume injected, calculated on the water velocity. As shown in this figure, for lower foam quality, the generation of stabilized CTAB stabilized foam, surfactant stabilized foam reaches the equilibrium in around 4 PV, and the corresponding to to 80 minutes. It stabilizes around 0.14 megapascal, which corresponding to 2 atmospheric pressure. The remaining water saturation drops to around 0.1. As the foam quality increase with the overall process becomes slower, and our hypothesis is that the foam generation is limited by the surfactant concentration, given a large number of bubbles forming at high quality. So when the nanoparticles were added to the surfactant, the overall process is less sensitive to the foam quality change. In all cases, the system reaches equilibrium within 1 PV. In this slide, the plot at the bottom shows the nanoparticle concentration as a function of a pore volume injected. As it is possible to see in these measurements, nanoparticles break through the column before 1 pore volume, indicating that at least a fraction of them travels with the gas bubbles. And then this slide compares the measured concentration of nanoparticles for experiments without and with foam. We can see here on the left-hand side, the nanoparticle breaks through around 1 PV when no foam is present. This means that they travel at the average flow velocity. In the presence of foam, they break through much earlier instead, and a large fraction of nanoparticles is absorbed onto the gas-liquid interface, traveling at the speed of gas phase, which is larger than that of water for high foam quality foams. Then this leads me to the conclusion of this talk. Here, a modeling and experimental work to study nanoparticle delivery by foam was presented. And we developed a model coupling foam transport, nanoparticle transport in porous media. And the measurement shows a faster nanoparticle delivery when foam is present, regardless of foam quality, which could increase to up to 86%. So the model agrees very well with the measurements and shows that a large fraction of nanoparticles is transported by foam, by bubbles, which are expected to inhibit nanoparticle agglomeration. So further work will focus on investigating the delivery of reactive nanoparticles in heterogeneous porous media and also in the presence of contaminants. So, and at last, we would like to thank the support from ACSPIF, NIE Fellowship of Stevens, and thank you for your attention.